Welcome back to the deep dive. So imagine you have a single sheet of paper, and your job is to fold that paper into a hypercomplex, self-sustaining 3D city.
With neighborhoods, infrastructure, specialized residents, everything.
Exactly. And that, in a nutshell, is the challenge of building the human brain. You're taking this early, kind of uniform central nervous system, the CNS, and you have to get every single cell to know exactly which neighborhood it belongs in.
Is it going to be forebrain, hindbrain, spinal cord, retina?
Right.
And that whole instructional process is called regionalization. For decades, studying this in humans was basically impossible.
Our models were just not there yet.
Not at all. We were stuck with animal models, which have really different brain architectures, or these, simple 2D cell cultures on a flat dish.
Which behave nothing like real three-dimensional tissue.
Exactly.
And that is precisely why this deep dive is so exciting. We are dissecting this massive systematic study that used human neural organoids.
The famous mini brains.
The mini brains, yeah. And this is the key point. These aren't just blobs of cells. These organoids can self-organize. They can mimic really complex structures, like cortical layering and even interneuron migration.
They're the closest thing we have to watching human brain development in a petri dish, no question.
But even with a great model, there was a huge gap in our knowledge.
A giant map was missing. We didn't have a comprehensive survey of how the developing human neuroepithelium, that's the foundational tissue, responds to the critical molecular signals that drive this whole process.
These signals are called morphogens, the architects of the brain.
Right. And the big question wasn't if they mattered. We knew they did. It was how the timing, the concentration, and the combination of these morphogens actually choreographed the final outcome.
So the mission here was just pure, massive-scale data collection. They basically built a robotic, high-throughput lab and systematically tested nearly 100 different chemical environments.
And their tool for reading the results was single-cell RNA sequencing, or cRNA-seq.
Which lets them see what every single cell is doing.
Yes. The goal was to map out precisely what recipe of morphogens, the when, the how much, and the which mix produced which final cell type after 21 days.
It's a monumental effort to replace guesswork with just concrete data. Hashtag deconstructing neural development and the experimental engine.
Okay, so let's unpack this by setting the stage a little. How do the initial cells even know they should become neural tissue in the first place? What's the very first decision?
Well, it starts with a classic molecular tug-of-war. If an early cell is exposed to signals like bone morphogenetic protein, BMP, it basically defaults to non-neural tissue.
To become skin or something else.
Skin, mesoderm, yeah. To switch into the neural lineage, to start building the brain, that BMP signaling has to be actively inhibited. And at the same time, the cell has to get activating signals like fibroblast growth factor or FGF.
So once they've committed to becoming a neural plate, they start forming the neural tube, and that's when it has to split into all the different regions. Forebrain, midbrain, hindbrain.
Right, and that's where the morphogen gradients come in.
The molecular GPS system. Exactly. Regionalization is defined by these unique morphogen concentration gradients running along two main axes. Anteroposterior, which is front to back, and dorsiventral, top to bottom.
So to recreate this in the lab, the researchers started with human stem cells. They aggregated them into these small spheres called embryoid bodies, and then started culturing them. What were the key molecular levers they were pulling?
They focused on modulating 4 major signaling pathways that are crucial for early development.
Okay.
So they tested activators and inhibitors for the WNT pathway, which generally guides posterior and dorsal development. They used signals like SHH or sonic hedgehog and FGF8B, which guide anterior and ventral development. And then they also tested things like retinoic acid or RA and different types of BMP.
And they weren't just dumping the chemicals in, right?
Oh, not at all. They precisely controlled 3 variables: short pulses for timing, finely tuned steps of concentration for the dose, and then, of course, many different combinations.
That just sounds like a mind-boggling number of experiments. How on earth do you track the fate of every single cell across 97 different conditions?
That's where the power of cRNA-seq comes in. If you think of a cell's identity, whether it's a neuron, a progenitor, whatever, as a kind of genetic fingerprint, scRNA-seq lets you read the full genetic fingerprint of every individual cell in that culture, not just an average of the whole population.
Which is so critical because an organoid is a mixture of all sorts of cells.
Exactly.
Yeah.
They analyzed over 100,000 single cells in total. And to make sense of all that data, they used complex data integration, basically comparing the genetic profile of every cell they sequenced against a known, established human brain cell atlas.
And what did that big map reveal?
Incredible diversity. The cells took on identities from, virtually every major brain region. Telencephalon, diencephalon, retina, hypothalamus, hindbrain, spinal cord, plus other related tissues like neural crest and even mesoderm. The immediate massive takeaway was that generating a pure homogeneous tissue is incredibly difficult. Most conditions created this wide array of cell identities.
So complexity is just baked in from the start. And here's a surprising detail for me. You might think that if you blast a cell with a super high dose of a signal, you'd get a simple, consistent result. Is that what they found?
It was actually quite the opposite. Higher morphogen concentration didn't necessarily simplify or homogenize the tissue. In many cases, as they increased the dose, it didn't just intensify the existing effect. It radically changed the cell's fate.
It created entirely new cell types.
Yes. It just rewrites the script completely. Hashtag, tag, tag, the precise control of timing and concentration.
All right, let's dive into some specific examples to see this sensitivity in action. Retinoic acid, or RA, was a perfect case study for this, especially with timing. What did they find about when RA hits the cell?
RA showed this dramatic time dependence. If the cells received RA at the absolute earliest time points, we're talking days 0 to 3, it didn't promote neural tissue at all.
It did the opposite.
It actively pushed the cells toward non-neural ectoderm. It basically killed the brain-building process before it could even start.
But what if they waited and supplied it later?
If they waited until days 9 through 20, RA became this powerful promoter of posterior fates, specifically retinal and hindbrain progenitors.
That's a massive switch, just based on timing.
It's incredible. Think about it. The same molecular instruction delivered on Monday means become skin, but deliver that same instruction on Friday and it means become an eye or the back of the head.
That sounds terrifyingly fragile. And I assume within that later window, the dose also mattered a lot.
Oh, absolutely. Low RA concentration promoted retinal progenitors, the future eye tissue. But as they progressively increased that RA concentration, the tissue became enriched for more ventralized and posterior fates.
So specifically hindbrain.
Hindbrain and posterior floor plate cells. You are literally dialing in the anteroposterior location of the resulting brain region just based on the concentration of a single chemical.
That's amazing. Okay, now let's look at the WNT pathway, which governs the posterior and dorsal parts of the brain. What happens when you inhibit that signal?
When they inhibited WNT signaling, especially early on, the cells enriched for telencephalic progenitors.
The telencephalon being the most anterior or forward fate, the future cerebral cortex.
Exactly. So turning off the go back signal pushes the cells all the way to the front.
And if you activate WNT, the go back signal.
That caused strong dorsalization and posteriorization. It led to the formation of neural crest cells, which, eventually become peripheral nervous system neurons.
And get this, the highest WNT concentrations actually pushed cells completely out of the neural zone.
Right. It induced non-neural fates like mesenchyme. It just proves there's a sweet spot, and too much signal is just as bad as too little.
What about the other major players, the BMP signals? We think of them as the non-neural switch.
And this study confirmed that role. Early supply of either BMT4 or BMT7 prevented neural specification. Early BMP7 was particularly effective. It resulted in nearly 90% mesenchymal cells.
So non-neural connective tissue.
Correct. But they also diverged later on. Later, BMP4 guided cells toward retinal fates, which makes sense because it activated genes associated with optic cup development. But BMP7, surprisingly, reactivated pluripotency markers in cells even when added later.
It's like it can reset the cellular clock.
In a way, yeah.
Let's finish this section with SHH and FGF8, the anterior and ventral guides. Were there any human-specific findings here?
Yes, there were. Both SHH and FGF8 induced telencephalic fates, but again, concentration was the key differentiator. As they sustained a progressively higher SHH concentration, it induced this beautiful transition.
From what to what?
The cells moved from telencephalic to hypothalamic progenitors. This perfectly models the natural spatial gradients of SHH in the developing human forebrain.
Wow, so the lab reproduced a natural gradient just by changing the dose. But you mentioned a difference compared to, say, animal models.
I did. SHH had much stronger ventralizing and anteriorizing effects than FGF8 in these human organoids. At high concentrations, it was generating hypothalamic floor plate cells.
And that's different from what we see in mice.
It is. This contrasts with common observations in mice, suggesting that FGF8 might play a less dominant role in human anterior brain patterning. And this is exactly why we need these human-specific models.
So the biggest lesson here is that early development is just this dance of exquisite sensitivity. A tiny shift in timing or dose of a single factor can completely redirect a cell's destiny. #tag decoding regulatory networks and morphogen interactions.
Okay, so understanding the outcome is one thing, but understanding the mechanism, the internal wiring, that's another. How did they move from just observing cell fate to figuring out what was happening inside the cell?
They dove into the genetics using the regulon concept.
Yeah, think of a regulon as a master switch. That's a transcription factor. And the entire bank of light bulbs it controls, which are its target genes. The researchers mapped which morphogens activated which master switches.
So which molecular switches were connected to which signal?
Well, signals like SHH and RA were linked to several common master switches. But what was truly insightful was seeing how dependent these switches were on timing versus dose.
Okay, how so?
For instance, the SHH and RA regulons showed this wide dependency on both timing and concentration. But the FGF8-linked regulons, they relied strongly on concentration only. And the BMP-linked regulons were surprisingly dependent almost entirely on timing.
Can you give us an example of that in action, that specificity?
Sure, look at SHH again. SHH controls two completely different developmental programs in the forebrain. The activation of one master switch, the NKX2-1 regulon.
Which is key for a region called the MGE.
Exactly. That required a high SHH concentration, but it was homogeneous across all the time points they tested. It just needed a lot of SHH, period.
So it didn't care when it saw the signal, just how much.
Precisely. But the activation of a different switch, one that's essential for thalamic development, required later time points and even higher concentrations.
Two distinct fates, driven by the exact same morphogen, but differentiated only by its kinetics.
Precise time and the precise dose.
This complexity just keeps deepening, especially when you start mixing the signals. What happens when you combine, say, WNT signals with RA, or SHH inhibitors with SHH activators?
They started probing these combinations to see if the effects were additive or synergistic. They used some sophisticated metrics, but essentially they were asking if signal A causes outcome X and signal B causes outcome Y, does A plus B equal X plus Y?
Or does it create some brand new non-additive outcome, Z?
And very often it was Z.
What was the most interesting finding?
A really clear synergistic effect in one of the stem cell lines. A combination of WNT inhibition and SHH activation led to a significantly higher proportion of non-neural ectodermal cells than you'd ever expect by just adding the individual effects together. This nonlinear synergy just pushed the cells unexpectedly far outside of the neural lineage.
It's like mixing two neutral ingredients and creating a powerful new chemical by accident. It tells us that really optimizing organoid development isn't just about dialing individual molecules up and down.
No, it's about understanding these complex chemical reactions happening inside the dish.
So far, we've focused on the signal itself. But the study's final, and maybe its most crucial, finding focused on the context.
The biological differences in the starting material and the culture environment itself.
Did the starting cell line matter?
It mattered dramatically. They tested 4 different human stem cell lines and 2 different neural induction methods. The standard minimal induction media and the very common dual SMAD inhibition method. The cell line and the induction method both profoundly impacted the final patterning.
That sounds like a reproducibility nightmare for scientists. For example, RA induced a specific region, the posterior floor plate, in 2 different cell lines pretty consistently.
It did.
But one of those cell lines, under low SHH signaling, also produced completely different tissues, hindbrain and retinal progenitors, while the other one just didn't.
That's the challenge. And here's the paradox of the dual SMAD method, which is generally considered the superior one. It promotes stronger neural fate and maturation. You get a higher ratio of neurons.
Sounds good.
It is, but this better differentiation method made the final patterning outcomes more susceptible to variability across different cell lines than the simpler control media.
So it makes them better neurons, but less predictable in terms of what kind of neurons they become.
That's a good way to put it. Improving one aspect maturation increased the difficulty in controlling the final regional fate. This inconsistency even reached the regulatory network level.
What do you mean?
Well, while morphogens usually followed the same general directionality across different cell lines, the base culture media could flip the script. In one stunning example, under the same WNT activator signal, the HOX regulons, which are critical for posterior identity, were activated in the dual SMAD culture but downregulated in the control media.
The culture medium itself completely changed how the genome interpreted the signal.
Exactly. Context dependency at its most extreme.
Finally, they compared how the geometry of the culture, the physical environment, influenced all of this. They compared a continuous SHH gradient delivered via a complex microfluidic chip to the static discontinuous steps they used in the 3D organoids.
And the results were starkly different. The tissue receiving the gentle, continuous gradient showed gradual, smooth patterning, a really nice transition from one forebrain region to the next.
And the 3D organoids.
The organoids getting these static, discontinuous steps showed radically different, much more ventral regions, hypothalamic floor plate cells, and they had these sharp, dramatic shifts depending on whether a certain threshold of SHH was met.
So why did the 3D organoid environment cause such an extreme reaction compared to the smooth, controlled environment of the chip?
Live imaging gave us the explanation. They saw that the expression of critical identity genes, like the marker for MGE formation, didn't just emerge uniformly across the organoid.
It appeared in spots.
It appeared at localized, foci tiny little hotspots that gradually spread, even though the whole organoid was submerged in the same chemical bath.
That sounds like a neighborhood reinforcing its own identity.
That's a perfect analogy. This suggests a powerful three-dimensional feed-forward mechanism in the organoid structure. Once a few cells start adopting an identity, their physical proximity and the lack of fluid flow cause neighboring cells to reinforce that initial commitment.
Which leads to a much more dramatic all-or-nothing fate shift.
Yes, compared to the relatively open two-dimensional cultures. And this is the crucial finding about why 3D organoids are such powerful but also complex models to work with. Hashtag outro. So this exhaustive study, analyzing over 100,000 cells, has provided an unparalleled map of human neural specification. It overwhelmingly reinforces one central truth.
Which is?
Regional specification is intensely sensitive to the kinetic properties, the timing, the concentration, and especially the environmental context and the physical structure of the molecular signaling.
And the sheer scale of this data is designed to be combined with deep learning models. We're moving toward a future where we can actually predict and optimize differentiation outcomes in these systems.
It replaces years of trial and error in neuroscience research. It's basically a roadmap for custom building human brain tissue.
Which lets us ask much more sophisticated questions about developmental disorders that might be rooted in these subtle signaling errors.
Exactly.
So to leave you with a final thought, we've seen that a tiny difference in culture medium, just switching between two common protocols, can completely flip whether a critical gene network, like the HOX regulons, is activated or suppressed by a single signal. Given that exquisite sensitivity and knowing that the 3D environment creates these self-reinforcing, dramatic commitments, what does this level of tight, context-dependent control tell us about the hidden robustness, or perhaps the terrifying fragility of human brain development as it occurs naturally in the womb? That is the fascinating vulnerability this new map reveals.